There we go. Hey, can you hear me, everyone?
Yes, we can hear you.
Great. And can you see my screen?
We see a empty desktop. There we go.
Okay, perfect. Thanks. So, hello, everyone. My name is Sam Gelman. I'm a PhD student here at UW-Madison, and I'm advised by Tony Gitter. And today I'm going to be talking about learning protein sequence function relationships from high-throughput molecular simulations. This is a Condor heavy project, and there are two ways we use Condor. One is to run molecular simulations with proteins, and that requires lots and lots of CPUs and thousands of compute hours. The other way is to perform machine learning, specifically to train neural networks. And that requires GPUs and long running jobs to train large models. So I'm excited to talk about how we use Condor and how the Center for High-Throughput Computing and the Open Science Grid have made this research possible. But what I want to do first is give a quick introduction to the research project. Why are we doing molecular simulations? Why are we doing machine learning? And then I'll talk about my experiences with Condor and the lessons I've learned. Proteins are large biological molecules made up of amino acids. They perform lots of important functions in biology. For example, enzymes help digest nutrients, and antibodies help fight viruses. And in general, proteins are very diverse, and there's a wide range of functions that they perform. Our goal is to do protein design, which is when we want to create or modify proteins to have a desired or improved function. For example, some laundry detergents contain engineered enzymes that make the laundry more effective and save energy. Another example is antibody proteins, which are being engineered to treat cancer and other diseases. To understand how we do protein design, we need to know a little bit about the protein sequence function relationship. Every protein is defined by a sequence of amino acids, and amino acids are smaller molecules that chain together to form the larger protein molecule. There are 20 different amino acids, and each has its own physical and chemical properties. Here, looking at green fluorescent protein, this is a protein found in jellyfish that converts ultraviolet light to green light. It's made up of 237 amino acids, and in general, a protein can be anywhere from several dozen to thousands of amino acids. And the amino acids interact with each other and fold into this 3D structure, and then that structure determines the function of the protein, which in this case is fluorescence. Now, if we were to replace one of these amino acids with a different one, that causes a change in the structure and function of the protein. So here I replaced the E that was in position 4 with an R. And this creates a variant referred to as E4R. This variant has decreased brightness because of the amino acid substitution. So one of the challenges with protein design is that it can be hard to know how an amino acid substitution will affect function. Um, if we want to improve function, what amino acid substitutions do we have to make? So what biologists can do is set up experiments where they synthesize these variants in the lab and then they measure their functional properties. So our goal is to use this experimental data to create models that generalize and predict the functional activity of any given variant. The experimental data consists of variants that have been tested in the lab, and there's an associated functional score telling us how functional that variant is. And what we do is use supervised learning to train a neural network to predict that functional score. And once the network is trained, we can use the trained network to make predictions for new variants and find those high functioning variants that we want to design and engineer. Now, one problem is if the training data is limited or biased, it can be hard for the network to generalize effectively. So this is the case with some existing data sets that are small scale, or they only contain examples of certain mutations. So to tackle the challenge of the limited experimental data, we're proposing an approach called METAL, which stands for Mutational Effect Transfer Learning. And the idea is to augment our limited experimental data with additional data from molecular simulations. Then with transfer learning, we can leverage the knowledge from the simulations and apply it to the experimental data. And then that should enable our models to learn from fewer examples and to better extrapolate outside of the experimental training data. So that's our approach, and our simulated data is coming from Rosetta. You may have heard of Rosetta before. It's a really popular protein modeling software, and it captures different aspects of protein stability. So the outline for this method is as follows. So first, we're running molecular simulations, which requires high throughput CPU jobs. Then we train neural networks on those simulations, which requires long running GPU jobs. And then finally, we transfer and fine tune the models on the experimental data, and that requires high throughput GPU jobs. So let's go through and talk about these stages. For the molecular simulations, to give you an idea of scale, we have tens of millions of protein variants that we want to model with Rosetta. And those variants can all run independently from each other. So it's perfect for high throughput computing. And I split the variants into hundreds of thousands of jobs, and I target about 5 to 10 hours of runtime per job. I think sometimes it can be hard to estimate how long a job might run because it depends on a lot of factors. For this project, I wrote a function to estimate runtime based on the size of the protein. Then I have a simple greedy algorithm to balance the jobs at around 5 to 10 hours. Something that went really well was just how much capacity there was, and there is. Especially with Open Science Grid, I was running thousands of jobs simultaneously. A challenge was that there were some bad servers and sites, actually, that kept scheduling my jobs, and then the jobs would crash. So I was able to mitigate some of this by blocking some of the servers. Also, I used a Condor feature called on exit hold, which puts jobs on hold if they exit with a certain error code. And I set up my jobs to exit with this error code. And so instead of being finished as crashed jobs, they went back into the queue. And I used auto release to get those jobs running again. And they usually got scheduled on good servers after that. Moving on to the machine learning part of this, we're using transformer networks to model the protein sequences. And if you haven't heard, transformers are all the rage right now, especially in natural language processing. But transformers and deep neural networks in general can take a long time to train, and they need GPUs. So I want to talk about getting GPU resources, and then checkpointing, which I found to be very important, and as well as logging and monitoring your job's progress. So we use GPUs from several different sources, and the cool thing is that with Condor, we have a single submission point to access those sources. Our main resource is the UW Grid, which includes GPUs from my lab group as well as others, and as well as the GPU Lab, which is a great GPU resource here on campus. And in the past, though, we've needed additional capacity for some high throughput runs, and we've been able to access other clusters on AWS and the Argonne National Lab via HTCondor, so it's been very convenient. Moving on to checkpointing, checkpointing is basically required for long-running models because a lot can happen when you're training a model for a couple weeks. You could hit runtime limits, you could get evicted, or your job could just crash. And when that happens, you don't want to lose the training progress, right? So there are really two aspects to checkpointing. One is saving and restoring the not just the model weights, but also the optimizer state and any other um, parameters that you have. And the other aspect of checkpointing is actually integrating with the HTCondor checkpointing system, which is um, which requires certain error codes and certain flow. So PyTorch Lightning is a framework that's built on top of PyTorch, and it has some features that I found to be really useful for checkpointing. Out of the box, it can take model checkpoints that contain the model weights and the optimizer state and everything else you need to resume a model. So that's awesome. There's also an excellent callback system that lets you add functionality in a modular way. So with HTCondor checkpointing, um, your job needs to exit with exit code 85 regularly, right, in order to get Condor to perform the checkpoint. Obviously, this is not built into PyTorch Lightning, but I was able to write a custom callback that exits the script with code 85 every epoch, or really, I set it up to do whatever interval I want. And then I was able to integrate that really easily with the full model training framework. So PyTorch Lightning really enhanced my Condor experience in this case. Logging. Okay, so we can submit long-running jobs and we can checkpoint them, but how do we keep track of the progress? And sure, you can use the Condor tail command to access your job's std out, and maybe you're printing some useful status updates every epoch or something like that, but that's fairly limited. And we need a better system to monitor the training progress and the training results. So for this, I found that weights and biases is also really enhances the Condor experience and the machine learning experience. It's Weights and Biases is a cloud-based machine learning platform, and what I do is the execute nodes log directly to Weights and Biases over the network. So Weights and Biases takes the incoming data and it creates pretty graphs and charts, and it gives you a really good idea of how the model training is going. And it also tracks system utilization, so you can see how much your GPUs are being utilized, how much GPU memory you're using, and all that good stuff over the entire duration of your run. So I found that this has been really helpful for training models with Condor because it lets you catch problems early. You don't have to wait for the model to finish before seeing, hey, that the training's not actually going that well, and maybe we need to run it with a different hyperparameter. And another cool thing is that it combines logs from multiple runs into one cohesive view. So when I'm using Condor checkpointing and I'm constantly stopping and restarting my job, the logs still are as if it was just running for one job without stopping. So as a refresher, our objective is to predict protein functional scores based on experimental data. And to overcome the challenges of limited experimental data, we are creating our own data using molecular simulations. Then we perform transfer learning from the molecular simulations to the experimental data. And we tested this approach as well as several baselines. And this graph shows the performance of the final models with varying training set sizes. So our approach is represented by the green line on top. That's called METL, METL, what this graph is showing is that this transfer learning approach generalizes better with smaller training sets than any of the other approaches we tested here. So we can really see the benefits of all those hours of molecular simulations and the transfer learning. So this work I talked about today is still in progress, but if you're interested, our previous work is covering some of the same topics, and you can check out that publication. We actually collaborated with biologists to design improved proteins using our models, and there was some moderate success there. So in conclusion, thanks to HTCondor and the Center for High-Throughput Computing and the Open Science Grid for making this research possible. Without it, we would, I don't even know if we could do this because we just use so much compute resources, and it would be really like cost prohibitive to do it on a different cloud like AWS. Um, so thanks to everybody who helped with this research, and thank you for listening. So I'm happy to take questions.